Starting chapter 8 for microbiology, this would be metabolism. All right, metabolism is just all chemical functions that occur inside of a cell. Doesn't matter what kind they are. If it's a chemical reaction, then it's metabolism. We break it down to two different types of chemical functions, though. We can. One, catabolism. This is taking a larger molecule and breaking it into smaller pieces and releasing energy out of it. So an example would be taking a protein and breaking it down to the amino acids. That's going to release the energy as you break those bonds and make smaller pieces. The opposite of this is anabolism, and that's where you take small ones and put them together to make a larger molecule. This requires energy, so you have to put energy into it. And it's just the reverse of the process. So you take amino acids, put them together to make a protein, and well, it takes energy to build something. So catabolism and anabolism. Enzymes are very important for life because they speed up chemical reactions without raising temperatures. So they do, that's what a catalyst does. It's going to make a reaction occur more quickly. So we call this the process of lowering the activation of energy or energy of activation. In other words, it means it takes less energy to do the same reaction with using the enzyme than without the enzyme. These enzymes are not permanently altered. They can change shape a little bit while they're doing the process, but when they're done with it, they come back to the original shape. And they are specific for reactions, for specific substrates. Substrates is what you're going to get out of the reaction. So again, here they're showing you for a reaction to occur fast enough for life to occur, you either add a lot of energy into it up here, or you use enzymes, which lowers the amount of energy it needs to get the same product. What is an enzyme? An enzyme is really a protein, all right? And they have some fancy words for them here. So the apoenzyme is the protein portion of it. And then you have some non-protein portions that are on, and those are the cofactors. And the combination of these are the haloenzymes. So they're showing you the purple here is the enzyme, and then they have the cofactors onto it. Usually they're vitamins or something along that line. These are some common ones. Enzymes, notice that they all end in ASE. Those are enzymes. This is what they do, and this is the cofactor that helps them out, which is why you need to have your vitamins and minerals. Enzymes are proteins. Again, proteins are simple amino acids put together in chains, and they're folded up. So this would be a primary folding, secondary, tertiary. And when you have a couple of them together, you call it quaternary, or we call them globular proteins at that point. Each enzyme has a specific active site. So there's one spot on that protein that is going to be active for whatever reaction it's looking for. They're showing you here, they are specific. This particular substrate can fit into this enzyme, that one can't. So that way each enzyme is only doing one chemical reaction. Again, we talked about the micronutrients. Those are your coenzymes, your vitamins and minerals. I'm not going to go into a lot of detail on that. I just know that they just help enzymes work better. Exoenzymes, these are enzymes that are outside of the cell. For a human, a good one would be the amylase enzyme in your mouth. It's the second one. It breaks down starches into sugars, but it's just in your saliva, so it's outside of the cell. And then most enzymes are endoenzymes, where the substrate gets brought into the cell, and the enzymes are in the cell reacting. But they're inside the cell, so they're called endoenzymes. Again, here's just some more enzymes, what class they are, their substrate, and what they do. Constitutive enzymes. These are ones that are always there, no matter whether we have a substrate, or we need the product, or whatever. They're always going to be produced. Regulated ones are ones that we don't have a set amount present, so they're either regulated by gene control or by the amount of substrate that is present. If we have more substrate, then we produce more of them. In other words, some examples we'll get to in a moment. Reactions that these things are occurring. So if you add water, if you remove water from it, all right, then this is a synthesis, a condensation. All right, you're taking these two, and you're removing water from this. So you pull water out of the reaction. On the other side, if you add water to it, so you're taking this water molecule, you're breaking it down, you're going to make two different ones. This is catabolism in this case. Over here was an anabolic, because you're taking two small ones and adding them together. So for the most part, they work that way. Again, you're in hydrolysis, you add water to the reaction, and in condensation, you remove water from the reaction. So an enzyme. Well, we didn't go over this, but enzymes do not become part of the final product. They are specific, or it is specific for a certain substrate. It is not consumed by the reaction. It is not only a protein, remember the cofactors. So the only one of these that is true is that one, they can be outside of the cell. The other four are false. The environment's going to affect these main things. There's temperature, pH, and osmotic pressure, but temperature is the big one. As you heat up remember, enzymes are proteins. If you heat up a protein, they'll change shape. And if they change shape, then they no longer function. And pH can do this, and again, osmotic pressure can affect this. So those are the main things for affecting enzymes. Liable means that they're unstable, it just means that they're easily broken down or easily denatured. And again, denatured is where they break these bonds and it changes the shape. Again, if it changes the shape, it doesn't work. Example I like to give is egg white, the clear portion of it. When you crack an egg open, it's clear, but as you add heat to it, it changes to white because you denatured, you've changed the shape of that protein. And once it's changed, it cannot go back. So that's why egg white, even as you cool it off, it doesn't become clear again. These are some metabolic pathways. Linear means you have the first reaction, you get a product from it, then you have your second reaction, you get a product from it, third reaction, and so forth. And that the product leads into the next reaction. Cyclic is the same thing, except you're going in a circle with it. The product here is going to be for this reaction, and its product is going to start that reaction, and so forth, and you keep just going. And you have divergent, convergent. Competitive inhibition. This is where, like I said, two substances will be similar in shape, and they'll compete for the active site on the enzyme. One of them is the actual substrate where you're going to get your product, and the other one is not. We have a picture coming up of this. I'll just go to right there. So these two, they're partially the same shape, and if one of them gets in, if the correct one gets into the substrate gets in, then your reaction continues. If the other one gets into it, it does not continue with the reaction, so you don't produce anything. So these two are competing for the same spot. Non-competitive reactions or enzyme inhibition. These are where they're going to be regulated either by the making, which is the genetic level of it, or by what substrates there, or some of the product even. So again, this is competitive. This is both competitive also. This side is, I apologize. Comes in, you have your substrate, it goes in, you have your reaction, you make your products. One of their products will come in and inhibits the reaction from going on. So that way you don't get too much of the product. And as that product gets lower, then you'll make more. The gene regulation of it here, since we have to go from DNA to RNA to protein, then it gets folded up and makes our enzyme. We have our reactions, we get our products. This product will then go and stop the copying of the DNA. It can stop it from going from DNA to RNA or from RNA to a protein. And if we stop it anywhere, then we don't get the enzyme. And this is a way to regulate the reactions. This is a chart in your textbook. This one is very good for understanding enzymes. Again, they're mostly a protein. They're an organic catalyst. In other words, they speed up the rate of the cellular reactions, so the reactions become fast enough for life. And they do this by lowering the activation energy. Each enzyme has a specific shape and specificity and function. They don't do their one thing, and that's it. They have the active site. They are much larger than what the molecule they're dealing with. They do not become part of the product. They are not permanently changed by the reaction. So they get recycled. They can be affected by temperature and pH. And they have some ways of giving you some feedback. Energy. Energy is just the ability to do work. All right? The forms of energy, we have the thermal, radiant, electrical, mechanical, atomic, and chemical. Most of these are not compatible with life. I mean, we do have microbes that can deal with a lot of these. But chemical energy is the best one for life. And most biological chemical energy reactions occur naturally, just at a slow rate. And so that's why we need the enzymes to make those rates occur faster, so that life can happen. Because we don't want to get too warm. We don't want radiation, because radiation damages DNA. Electrical is not practical. Atomic's not, well, this radiant here is light. So I should say some organisms are using that. And then we have radiation. So again, chemical is the best one to access energy from to perform our metabolic functions. All right. Before we were talking about the substrates, and we were talking about that catabolic and anabolic. Now we're just talking about the energy. And we classify these as two different types of reactions. Endergonic, which are ones that we have to put energy into it. So this would be along with the the anabolic reactions, and our exergonic reactions, which release energy. So these go along with our catabolic reactions. These terms here are used to describe the flow of the energy. Catabolic and anabolic are used more about the building or breaking down of the molecules. Biological reactions are reaction and reduction and oxidation reactions, redox. Easy way to remember them is oil rig. So O is an oil for the oxidation, and the L in it loses. So oxidizing loses electrons, and then reduction, and then the G at the end, gains. So if it's an oxidizing reaction, it's going to lose electrons. If it's a reduction reaction, it's going to gain electrons. So if you look right here, the hydrogen molecule here, in this reaction, it's going to give up, it's going to give up its enzymes to the oxygen molecule here. So it's an oxidation, so it loses electrons. And in this case, this oxygen molecule right here is going to pick up those extra electrons, so it's going to be reduced. Right. It's going to, it's going to gain them. So that's oil rig. That's the easy way to understand it. Now, for all this energy that we're trying to get from molecules, we need a way of transferring them around. main ones that we're going to talk about are NAD and FAD. Okay, in this course, there you can get a little more technical into the NADP and the coenzymes, but we're going to focus on the NAD and FAD. And this top one here, this one is NAD, and then we're going to have another one. Right here, our adenosine triphosphate, or ATP. This one is the main one. If we come back up here, NAD and FAD, these are just going to carry some energy until they can be made into ATP. And ATP is what all organisms will use for energy molecules. Let's come back to this question. If a molecule has been reduced, it is going to gain electrons and hydrogen. ADP, again, is the currency. All life uses it. 3 parts. You've got your adenine, your sugar, and your phosphates. These are high energy bonds. Usually we just talk about the third one being broken off, but we can break off the second one. And based on the number of phosphates, they have different numbers. So triphosphate, diphosphate, monophosphate. This is the first reaction in glycolysis. This is the first enzyme in it, hexokinase. It's much larger than its molecule. So the product is, the substrate is glucose. So this is how we get energy. Get your glucose is in there. We take ATP. We're going to break one of those bonds and add a phosphate to this glucose molecule. So see ATP to ADP. And then we have a substrate, a product, and the product will go to the next line. This is phosphorylation of glucose, because we are adding a phosphate to the glucose. Again, this is going to be the first step in glycolysis. There's a few others of these. Most of the ones that we're going to be talking about are going to be substrate level and oxidative phosphorylation. I don't go into photosynthesis very much in this class, but again, it's just about adding a phosphate. So how do we get energy for life? 3 main pathways for aerobic pathways are glycolysis, Krebs cycle, and then the respiratory chain. Aerobic respiration is using oxygen. Anaerobic respiration does not use oxygen, uses something else. And then fermentation does not do the Krebs cycle or the respiratory chain. It just stops with glycolysis. So if you look through these processes right here, the aerobic, this is the one we're going to focus on. This one is very similar, just different final electron acceptors. And then the fermentation, it just does glucose. So everything does glucose, all life. So over here, we can, if everything goes perfect, we can make a microbe can make up to 38 net gain of ATP using the aerobic pathways. Anaerobic, it depends. And then with fermentation, they only produce 2. Does not produce a lot, but it is faster because it's only doing glycolysis. But we want to produce a lot of energy for life, so that's why more things go with these pathways. All right, glycolysis is glucose and lysis. So we're breaking a 6-carbon glucose down into 2 3-carbon pyruvic acids, and we are going to make some NADH. We're going to get in more detail with it. TCA is also known as the Krebs cycle. The TCA stands for the tricarboxylic acid, also known as the citric acid cycle. It's going to take the pyruvic acid from glycolysis, make 3 CO2 molecules, because see there's 3 carbons for each one of them, and it's going to produce some NAD and FADH. Now you'll have to do this twice because we're making 2 pyruvic acids up here for every 1 glucose molecule. Then the electron transport chains, where we're going to take the NADHs and FADHs, and we're going to do the oxidative phosphorylation and produce ATP at this point. All right, this is glycolysis. Some key things I want you to remember in here. So we'll take that 1 6-carbon molecule. In reaction 1, we are going to add an ATP. And in reaction 3, we are going to add an ATP. So far, we've only added energy to this molecule. At this point, we are going to break it into 2 3-carbon molecules. So from this point on, we must go through this cycle twice to explain everything. Once for each 3-carbon molecules. That's step 4 rearranges it a little bit. Step 5 is where we first get some energy out of it. So reaction 5, we're going to produce an NADH. At step 6, we are going to produce an ATP. I say a step, I should say reaction. And at reaction 9, we are going to produce an ATP. Now that's for 1 3-carbon molecule. Remember, we have to do that twice. So based on this, we add ATP at reaction 1, reaction 3. At reaction 5, we gain technically 2 NADHs. Reaction 6, we're going to gain technically 2 ATP. And at reaction 9, we're going to gain 2 ATP. And our final product is going to be that pyruvic acid. It's just going to lead to the Krebs cycle or fermentation, depending on what species we're talking about. For every reaction, there's going to be 1 enzyme. So we have 9 reactions, but we end up with 10 enzymes because there's a 10th enzyme in here that's rearranging the shape of a molecule. So these are some key things I want you to know about glycolysis. It consists of 9 reactions, but there's 10 enzymes. We have to use 2 ATPs to produce a total of 4 ATPs and 2 NADHs. That gives us a net gain of 2 ATPs. So if we back up right here, glycolysis makes 2 ATPs. Technically it makes 4, but we got to replace the 2 we used. Keep that in mind. And then the final product is going to be the pyruvic acid. The first product here is the substrate is glucose. The first enzyme is hexokinase. Come back up here. There it is, hexokinase. There's the first enzyme. And that is glycolysis, a brief overview of it. All right, so see here, glycolysis making the pyruvic acid. Then depending upon where we go, what's going to happen. We're going to stay with the respiration here. Now we are up to the Krebs cycle or TCA cycle. So the pyruvic acid is going to react with acetyl-CoA, a coenzyme A to make acetyl-CoA. What happens, you're going to produce 1, remember for each pyruvic acid, there's 3 carbons. You're going to produce 1 molecule of CO2 and 1 NADH in this, what we call lead-in reaction. Because it's technically not part of the Krebs cycle, of the cycle itself, but it is. So there's 1 item right there, that's the lead-in. Now we're going to there's 2 carbons left, because remember we took 1 off. We're going to combine it with this 4-carbon molecule over here called oxalacetate or oxaloacetic acid and make another 6-carbon molecule, citrate. And that's why this is also called the citric acid cycle. And you see right here the 3-carbon, the tricarboxylic acid. So that's where they get some of these names from. So there's, not including the lead-in reaction, there are 8 reactions in the Krebs cycle right down here. And most of it's just rearranging molecules. So reaction 1, you're going to produce this. Reaction 2, we're producing this. Reaction 3 is where we're going to release another carbon dioxide molecule and another NADH. Reaction 4, we release another carbon dioxide and another NADH. So there's our 1, 2, 3 carbon dioxide molecules that we release from 1 pyruvic acid from that 3-carbon molecule. And since we make 2 pyruvic acids in glycolysis, we must do this twice for every 1 glucose molecule. Then we come in over at reaction 5, we'll produce an ATP. Reaction 6, we produce an FADH. Reaction 7, we're going to add some water to it, and then in reaction 8, we're going to produce another NADH. So for 1 pyruvic acid, we'll get 1, 2, 3, 4 NADHs, 1 FADH, and 1 ATP. And since we have to do this twice, we double all of this. So we have a total of 2, 4, 6, 8 NADHs produced, 2 FADHs, and 2 ATPs. That's from the lead-in and the Krebs cycle alone. Then we also got to remember what we produced with the Krebs cycle, or excuse me, the glycolysis. All right. Now that we've done this, all those NADHs and FADHs will go to the electron transport chain, and this is where they're going to produce ATP. The ATP is produced in, by far the majority of it is produced in the electron transport chain. In eukaryotes, the electron transport chain occurs at mitochondria, and it's there's the cristae on the inside of it. As the membrane, what happens is the NADH will go through a series of reactions, breaking off the H's, those hydrogen ions, and they produce what's called a hydrogen ion proton motive force. And these ions will pass through this enzyme called ATP synthase and produce ATP. FADH does a very similar thing, but it doesn't go through quite as many reactions. I'm not going to break down these reactions on here. Just know that on eukaryotes, this is going to occur on the membrane, because most reactions occur on membranes of the mitochondria. All right, so there's that proton motive force where it's actively pumping these hydrogen ions out, and then they diffuse back through the ATP synthase, and they make ATP. On a prokaryote, they don't have mitochondria because, well, they are mitochondria. So they have that periplasmic space between the cell wall, and the plasma membrane. That's where they pump their hydrogen ions, and they have ATP synthase, and it goes through the similar process of making the ATP. Now, the final step is the oxygen being involved in this, and the oxygen is going to collect 2 electrons and make water. So, review all of this. Glycolysis takes 1 6-carbon glucose and makes 2 pyruvic acids. It uses 2 ATPs, so it has a net gain of 2 ATPs and 2 NADHs. The lead-in step produces 1 NADH per pyruvic acid, so it's going to produce a total of 2 of them, and it's going to also make 2 carbon dioxides. The carbon dioxides are just keeping track of where the carbon molecules are. The actual Krebs cycle here, remember, goes through it twice, just like the transition, it goes it twice because of these 2 pyruvic acids. We will produce, because it does go through it twice, we produce 4 carbon dioxides. See there, 2 plus 4 is our 6 that we had right here at the beginning. We're going to produce these 6 NADHs because it does it twice, 2 ATPs, and 2 FADHs. Now, you might be looking at this and wondering where it's coming up with these 34 ATPs. And what it is for every 1 NADH, you can produce 3 ATPs using the electron transport chain. For every 1 FADH, you produce 2 ATPs. So if we count them up, we have 2 ATPs here and 2 NADHs, and each one of those are going to produce 3, so there's 6, so we have a total of 8 right here. 2 NADHs, that's going to be another 6, so 8 plus 6 is 14. 6 times 3 is 18, so 18 plus 14 is what, 32? Plus 2 more here is 34, plus two FADHs, which produce 2 each. So there's another 4. So there's actually 38. And if we go way back here, 38 is ideally what it's going to produce. What this is showing you, those 34 ATPs, that's the ATPs made from the NADHs and FADHs, because we've already got 2 ATPs here, 2 ATPs here, those are the 4. So you add those other 4 on there, there's your 38. Notice that most of the carbon dioxide is produced in the Krebs cycle, and water is produced in the electron transport chain. So let me come way back here again. Got to come back quite a bit to the cellular respiration. Your 6 carbon molecule, glucose, plus oxygen is going to make 6 CO2s. And that should say 6 H2O. I didn't realize it didn't say that. Let's see if I can fix. Yeah, it should say 6 H2O. And that's this is a simplified version of how we get energy from all of these reactions. Remember, there's 9 reactions in glycolysis. There's 8 through Krebs cycle, plus another one in the transition or the lead-in reaction. So technically there's 9 here also, so up to 18 different reactions to do what we show you with cellular respiration. And that's not including everything that goes on in electron transport chain, and that just depends upon which molecule you're using, FADH or NADH. So it is a complicated process, and we're trying to really simplify it for you. Remember, again, the final oxygen is the final electron acceptor in aerobic respiration. So again, here, which part of aerobic respiration releases CO2, or should we say most CO2 is going to be glycolysis or Krebs cycle. Anaerobic respiration, without oxygen, goes through pretty much the same thing. You have glycolysis, Krebs cycle, electron transport, but they use a different molecule, like nitrate, nitrite, sulfate. And because of this, it's going to vary with whatever molecule they're using on how much ATP they can produce. Still, the maximum is going to be that 38 net gain, I should say, 38 net gain. Fermentation does not do the Krebs cycle, so it can't do the electron transport. It just stops at glycolysis. So it's a small amount of ATP, but glycolysis goes pretty fast, so they can still produce a lot of energy. Most of these products are going to be like acids or gases or alcohols, and that's what that's what it does. So we can break. We go through, when our muscles start to burn, they produce lactic acid, but then we can recycle this lactic acid back up here and still get energy out of it. So microorganisms produce a lot of different acids. They're showing you here, they're showing you different organisms producing different things. Most of these products are industrial, but they come off that pyruvate, which again, remember, is the product of glycolysis. This is just kind of a rundown of it. Now, we say, if you remember back, it said theoretically can produce 38, and the reason why is because as we go through this, the organisms, our bodies too, will take these parts, pieces of them out to build things, amino acid proteins, fats, nucleic acids. What are they used for? And we can break these things down also, which is why we can break down protein. We can break down fat for energy, not just carbohydrates, which is what glucose is. But again, it depends on what the organism needs, right? If you're coming up, that's analogy, you're building things, you're going down, you're breaking them down, you're releasing the energy out of it. So that's why it's theoretically you can get up to 38. And that's what they have. Pyruvic acid can be converted into amino acids through amination, and that amination just means you're adding a nitrogen group here. We can convert these through deamination, breaking those energy molecules back off. Glyceraldehyde 3-phosphate, which is one of the products in reaction 4 in glycolysis. You can use to make amino acids, carbohydrates, fats. So again, this is all theoretical, and that's why we don't always produce the maximum number of ATP. See, amination is adding the amine. Transamination is moving it from one to another one, but the amine stays there. Deamination is removing that off of there. It's the same with all of these terms. Phosphorylation is adding a phosphate. Dephosphorylation is removing a phosphate. Same concept, just so as long as you understand the concept of it, you could put in any word and make it work. And that is the end of metabolism. If you have any questions, please email me. And this one's more kind of a complicated one. So take your time going through it, watch it a few different times, double check with your textbook. And again, email questions if you have any. Thank you.